Good morning, students. Hope you'll be fine. In continuation to our last topic, due to some technical error, I cannot continue my this lecture regarding the structure of the DNA, how it looks like this DNA, and what we will see this DNA. It is as given by the Watson and the Crick that structure is double helix structure of DNA, and what are the main points in this structure that this DNA helix is a double stranded. It is a double stranded. We simply write it as DS. If it is single stranded, then SS. It is a double stranded, and this is made up of nucleotides. These are the nucleotides. These are each strand is made up of these chains of nucleotides. This is one nucleotide, this is one other nucleotide. Similarly, there is another nucleotide. And what we will see, the two nucleotides जो chains हमारे पास है, they are antipodal to each other. What we will see, what do you mean the antipodal to each other? See this structure, see this chain. This chain, it is a carbon first, carbon second, carbon third, carbon fourth and carbon fifth. In this carbon first, उसके साथ हम देखते हैं there is a nitrogen base attached by means of the bond, that is glycosidic bond. Now this fifth carbon, it is what we call as this attached with the phosphate group, that is by means of a bond, that is phosphodiester bond. Again, what we will see this at C. Here we will see this carbon 5 is free. Now this carbon 3, it has got the hydroxyl group here. And this is one of the difference between the ribose and the deoxyribose sugar. In case of the ribose, at carbon 3 and carbon 2 and carbon 5, there carbon 2 and carbon 3, there is the hydroxyl group. While as in case of the deoxy, at carbon 2 we have got the hydrogen, and at this we have got this OH at 3. Now this carbon third, it is attached, this hydroxyl group जो हम देखते हैं, this C5 of the second nucleotide, carbon 5 and carbon 3. Here we will see this phosphate group at carbon 5, it is attached with the hydroxyl group of the previous, what we call as the hydroxyl group, which is placed at, which is bonded at carbon third. Now we will see in this, if I draw the line like this first, what we'll see in this strand, ऊपर वाला जो है carbon 5, it is free, and this carbon 3, it is free. It is free. That is the single strand. What I have written here, this red one, this is the fifth carbon which is free, and the other end of it, that is the third carbon which is free. Now these two are parallel to each other. That means if these are the two parallel strands, they are parallel to each other. They do not meet any point. They are equidistant from each other. उसकी वजह यह है कि that this nitrogen bases, these are there, this adenine and thymine, or this guanine and cytosine, they are at equal distance from each other. They are at equal distance from each other. Second one जो आप यहाँ पर देखोगे, जो adenine and thymine, they are joined together by the weak hydrogen bonds, and this is a double bond. Again, guanine and cytosine, they are joined together by the weak hydrogen bond, but here it is the triple bond. Now anti-parallel means, now we have one more जो हमारे पास strand है, the strand which is in front of this first strand, previous strand. This strand, इसके सामने हम क्या देखते हैं, this is the first strand. अब इसके सामने हम क्या देखते हैं कि एक और strand है which is in front of the first strand, जो इस पहले strand के सामने है. अब इसका हम देखते हैं कि इसका पहला ऊपर वाला जो फ्री एंड है, that is carbon 3, and जो इसका bottom है other end, वहां पर हम देखते हैं जो फ्री है कार्बन, that is carbon 5. See here, it is a carbon 5 जहां पर phosphate है, and this one, that is the carbon third which is free. So this is called, this means the anti-parallel. They are parallel to each other, but in the first strand, it is the carbon fifth which is free, and other end of this strand, that is the third carbon which is free. In front of this strand, what we will see, the upper जो है, that is the third carbon which is free, and at at its other end it is the fifth carbon which is free. दूसरी बात जो हम ये कहेंगे, अब जो दिस DNA structure जो हम देखते हैं हेलिक्स structure, it has got what we will call as the same diameter. It has got the uniform diameter, and this diameter has been found about is equal to 20 angstroms. This is equal to 20 angstrom. Of why it has got the same what we call as this diameter or uniform diameter? Because of this distance between the nitrogen bases, Z base, guanine and cytosine, adenine and thymine, they have got the same bases. Second one, now one term समझाने के लिए if we take this हेलिक्स structure, इसको हम compare करेंगे by means of this spiral staircase. अब जो first step होगा इसका staircase का, it is a spiral. अब मैं यहाँ पर पूछूंगा जो वाला step and this step, when जो consecutive step जो होंगे first and that second, which are in the same line, which are parallel to each other, what we will see, that is called as one turn. ये वाला step it is spirally, ये spiral shape में है, and अब ये वाला step जो इसके सीधे होगा, that forms the one turn, and one turn is equal to 34 what we call as the angstrom. See this like this, it is equal to 1 angstrom. What we will see, this distance, it is equal to 34 angstrom, and each what we call as this, in this turn there are 10 base pairs. यहाँ पर मैंने दो ही बनाए, but in each turn there are 10 base pairs and each has got 3.4 angstroms. वो हमने किया 34 divided by 10, 3.4 angstrom जो उनकी distance है. This and this, the difference between these two consecutive, what we will see, these nitrogen bases, that is 3.4 angstroms. Watson and Crick has again showed us that there are various types of the DNA. We have got DNA, 5 types of DNA, DNA A, we have got DNA B, we have got DNA C, we have got DNA D, and we have got DNA Z. जो ये हमारे पास है, what are these DNA, उनके हम स्ट्रक्चर हम डिफरेंट आगे पढ़ लेंगे, बट what we study, this जो हमने स्ट्रक्चर बनाया, that is the DNA B, because in this DNA B एक जो हमारे पास peculiar characteristics है, that there are 10 nitrogen base pairs. There are 10 nitrogen base pairs which is also seen in this DNA B. This type. Second, if we look, this is the double helix. If we look this DNA from the top, अगर हम इसको ऊपर से देखेंगे, we will find that it is what we call as a spiral shape. It is spinning rather. It is spiral shapes, and what we will see, those turnings, those spinning जो होगी, it may be either clockwise or it may be either anti-clockwise. जब हम ऊपर से देखेंगे कि from the top of the DNA helix structure, we will see that there are turnings, and those turnings may be either clockwise or they may be either anti-clockwise. But in case of this DNA, ये हम सपोजन करेंगे कि that these turns they may be either, जो हमको देखता है कि that they may be what we call as this clockwise or anti-clockwise, but in case of this DNA double helix structure, those turnings are right-handed. They are clockwise. They are right-handed turn. So turn जो यहाँ पर है, it is in this manner. It is not in this anti-clockwise, but it is in this what we will call as clockwise, like this, not in this manner. It is in this manner, clockwise manner जो है, that is called as, that's the right-handed turning जिसको हम बोलते हैं कि, and that has got this DNA, it has got the clockwise turnings, and or we can say the right-handed turnings, and these turnings has been shown, या जो हमने स्ट्रक्चर पढ़ लिया, by the X-ray diffraction method, जो उस वक्त वाटसन एंड क्रिक ने किया, by the X-ray diffraction method, and by when the photos of those have been taken, we will find the structure like this. Now this was all about the structure of the DNA. In the next video we will proceed further. Until then, goodbye. God bless you. Thanks.